Scientists in Spain have developed a low-cost artificial cornea made from fish scales. The technology still has to undergo human trials, but the initial study results appear promising. We have this report now on how fish may allow humans to scale up their attempts to save people's sight. And a warning for the squeamish: you may need to look away in some parts of this story.
Believe it or not, your next cornea could start out as a fish scale. Researchers in Spain have created tissue that could serve as corneal implants using the scales of common market fish.
These tissues could solve problems, diseases and disorders in something as fundamental as the eyes.
Serious diseases in the cornea, the clear front part of the eye, are tough to treat because it doesn't have blood vessels and doesn't heal very well on its own. Most patients need a transplant, which involves removing the damaged cornea and replacing it with a healthy one from an organ donor. But donor corneas are in short supply. Thousands of people are living with sight loss because of this shortage. That's why researchers have been searching for alternatives, and they've found a promising one in fish scales.
The roughness of the surface is ideal for placing the corneal cells on top.
The team says the material has performed well so far, both in lab tests and in animals that received the implant. Still, it's a long way from being used in people, since human trials haven't begun yet.
It is highly compatible with implementation in humans and has theoretically yielded very good results in laboratory tests and animal studies.
It's a small step now, but it could lead to a big leap for people waiting to see the world again.
And I'm joined now by one of the scientists involved in that research, Professor Ingrid Garzón from the University of Granada. Professor, it's good to have you with us. Your study has developed artificial corneas from fish scales. Now, I'm sure if you were to ask someone walking down the street what they thought about fish scales and their eyes, they would have a squeamish reaction. How in the world did you come up with the connection between fish scales and being suitable material for corneas for humans?
Yeah, thanks a lot for your invitation. The idea came looking for a material naturally strong, transparent, and biocompatible. And these are the main characteristics of the fish scales, and even for cornea. So we were looking for very transparent material and very resistant. And these biomaterials, the fish scale, fulfill these minimal requirements for cornea models.
So let's go into that a little bit deeper here. How do fish scales become an artificial cornea? I mean, how complicated is this process?
Well, this process is a complex process that takes quite long in the lab. So first, we normally isolate the fish scale. Then we perform a process that is a dual process that is composed by decellularization to remove of the cells from the fish scale, the fish cells, and then we also have to eliminate the organic component of the fish scale that are on the surface. Then we have to do a functionalization of this biomaterial before to seed the cells to culture, before to culture the cells, the human cells on the surface of these fish scales. So it's a very long process.
Yeah, it's a long and complicated process. When could these fish scale corneas, when could they be tested in humans?
Well, for clinical translation, it's necessary to complete different important requirements for the European regulatory agencies. So normally, to move forward into the clinical scenario, we have to complete three different steps. The first step is the preclinical testing, the preclinical evaluation. We are in this study, we are performing this first step, the preclinical evaluation, that includes the in vitro and in vivo testing. And the second step will be the testing at the GMP facilities. And the third step will be to move into the clinical trial. So it's important to mention that we are in the first step. So we need further studies to move into the second and the third steps before to go into the patients.
Professor, can you give us a prediction? Is it possible to say how long these trials will likely take?
Well, we have a previous experience with three different products made from tissue engineering. That one is Cornea. It's a different model. The other one is an oral mucosa, and the other one is skin. So with our experience for those products that are now under clinical testing, it's about 10 to 15 years. So now we have a lot of experience in this, in all this regulatory and in all this process. So we probably can say maybe 10 or maybe less than 10 years.
And let's talk for a moment about why this research is even necessary. I mean, how do people end up with damaged corneas? Is it through disease, accidents, or congenital? I mean, what's the main reason for having a cornea that doesn't work anymore?
Yeah, unfortunately, corneal pathologies are among the leading causes of blindness worldwide, affecting more than 23 million of people around the world. So corneal pathologies can result from degenerative, dystrophic infections, inflammatory disease, or traumatic disease. So there is a huge list of pathologies and disease that can affect the cornea. So that's the main application of this technology.
And Professor, do we know, let's say that these fish scale corneas are successful in humans, will they be able to be used to treat anyone with a damaged cornea?
Well, these artificial cornea models are new products, as I said before. So the use may depend of the clinical approval of the European agencies and the regulatory agencies. So as researchers, we are studying how well they work in a long term and in different pathologies. So we may define this after complete the second step.
And Professor, let me ask you before we run out of time, I mean, let's be an optimist here. Let's assume that the testing and the trials are successful. What could these fish scale corneas, what could they mean for people who are losing their eyesight?
Well, it will make a huge difference because, as I mentioned before, there is a long list of donor scarcity through all these pathologies. So it's important for that people that can receive nowadays the transplants because they are waiting for a long waiting list. So this will make a huge difference because we expect that in the future we can store these artificial tissues into artificial bank, and all these tissues could be a solution for all these patients that are currently waiting for a donor.
Well, I'm sure all those patients, but everyone in general wishes you and your team all the success. This could mean a world of difference for a lot of people. Professor Ingrid Garzon from the University of Granada. Professor, we appreciate your time and your insights tonight. Thank you.
Thank you. Thanks a lot.